So the first thing we're going to look at is oligodendrogenesis. What is oligodendrogenesis? So oligodendrocytes are one of the glial cells, which are neurons in our central nervous system that are responsible for creating myelin. Now, they start off as stem cells, neural stem cells, and then they differentiate into something called OPCs. OPCs are oligodendrocyte progenitor cells. And these then need to be further differentiated into mature, remyelinating oligodendrocyte cells. It is only the mature oligodendrocytes that can create myelin, and they can only create myelin for a short amount of time. So there needs to be a constant regeneration of OPCs. So if we can prove that exercise can increase the number of OPCs, oligodendrocyte progenitor cells, then we can hypothesize that we're going to have an increase in remyelination. So forgive me again, I'm going to be looking at the article and going through important points and trying to explain it to you. So, here we go. So a study in 2014 by Mackenzie et al. Was looking at the role that myelin plays in complex motor skill learning. So mice were given access to a running wheel with irregular rungs as a skill learning task. So they have the wheel and there's irregular like little footsteps that they have to learn the skill to be able to navigate that. The authors found that running on the wheel was sufficient to increase the number of proliferating OPCs in the corpus callosum. So again, that's really important. The OPCs are the oligodendrocyte proliferator cells. If we have an increased number of those, then we have an increased number of the potential increase for a number of mature oligodendrocytes that can create myelin. And they found that this was an increase in the corpus callosum. The corpus callosum is a very dense white matter region in the middle of the brain that connects the two hemispheres so they can communicate with each other. So other studies have investigated the interactions between exercise and oligodendrocyte repopulation. The first study, which was in 2017 that we're talking about, examined whether exercise improved long-term memory deficits. Now this is important because in MS, memory and cognitive decline is very common, and hippocampal neuroplasticity in MS model mice. So the hippocampus is a region in the brain that's important for memory. So 10-week-old female mice did 4 weeks of forced treadmill exercise 20 days after they were caused to have MS. So what they found was when they were given the EAE induction, which is the MS, it decreased the hippocampal levels of MBP and CNPA. So these are both markers of that are responsible for the end-stage differentiation, turning OPC cells into mature oligodendrocyte cells. So when you have MS, you have a decreased ability to differentiate OPC cells into oligodendrocytes, and therefore a decreased ability to cause remyelination. So EAE mice, the article then went on to find that post-exercise, so 4 weeks after doing 4 weeks of exercise, there was a significantly enhanced expression of these two proteins, MBP and CNPAs, which are what? Responsible for differentiating OPCs into the mature oligodendrocytes. And that suggests that remyelination and oligodendrocyte repopulation had occurred. And also another interesting fact that they found was that in MS models allowed to exercise had a decreased number of apoptotic cell death in the dentate gyrus and the hippocampus. So what does that mean? So apoptotic cell death is like a pre-programmed cell suicide, and the dentate gyrus and the hippocampus are both regions in the brain responsible for memory. So this study shows that exercise in MS mice models had the potential, or did increase the number of differentiation proteins, turning OPC cells into mature oligodendrocytes, and it decreased the cell death in these memory regions. So it shows that there's a potential benefit of exercise for improving memory and cognition in MS patients, as well as improving oligodendrocytes and therefore remyelination. So another study investigated the effects of prior exercise. This is really interesting. Either high intensity interval training or low intensity continuous training on oligodendrocyte loss in young male mice after cuprizone demyelination. So this isn't the complete MS model of mice. This is the model of mice where they cause demyelination in the brain, not the spinal cord. So yeah, again, so there was 4 weeks of forced treadmill exercise, and then they were introduced to the toxin that caused the demyelination. So they did exercise for 4 weeks prior, and then they exercised for 5 weeks after. So as soon as they were induced with a demyelination toxin, they exercised for 5 weeks. And they found that cuprizone treatment decreased oligodendrocytes in the dentate gyrus and hippocampus again, but this was reduced by high intensity interval exercise, but not as much by low intensity continuous exercise in the dentate gyrus. However, in the hippocampus, there were similar benefits from both. So this is really interesting because it shows that higher intensity exercise potentially is superior to low intensity continuous exercise in providing improvements in CNS outcomes. Again, the 2 regions were looked at, the dentate gyrus and the hippocampus, which are responsible for memory. So we're gonna, just for the sake of time, I'm gonna go on to the next part, because they label about at least 5 to 10 studies per point. I'm just going through a few of them to highlight my point that exercise can improve the actual disease state and what's happening on a cellular level.